Yeah, cool. So today, thanks for having me. I'm going to be talking about sort of controlling generative models, in particular flow models in continuous spaces. I sort of came to this because, well, I made a lot of flow models in different domains, and then, well, the problem is that they're not very useful unless you're able to, do what you want. So, I guess we train flow models over many different domains, right? So images, for me, cells, so how cells change over time, for proteins where you're trying to model structure and trying to understand how you go from noise to structure of proteins, and then for molecules, right, still trying to understand maybe the dynamics or how the conformations are. And so in all these different domains you're able to train if you're given enough data what the sort of flow models look like. But sort of the open questions are, well, once you've trained on your data, what do you do with your model and how do you make it do what you would like? So today, I'm going to talk sort of about one flow model in proteins in particular, and then sort of different ways to think about controlling these and what are the requirements in different spaces. So just kind of talk about flow matching on proteins for a second. So here it's kind of interesting because we have very special representations for proteins, and they're on manifolds. And so here we use sort of a SE3N manifold, and the goal here is to sort of transport proteins from sort of a noise distribution to the 3D structure. And there's many different ways of doing this. And so here's three different versions, sort of a base model, which is sort of how everybody trains things now, with flow matching and diffusion, which is just sort of you take a random noise point and a random data point, you pair it up, and you sort of learn a model that can go from the noise point to the data point. And if you average this out over many noise data samples, then you get something that, given any noise points, you get a data point back, right? But of course, this really just gives you your data set back, unless your model is underfit, which of course all of them are, but you'd like to be able to control where and how they're underfit. And so there's sort of different ways to parameterize this, but that's sort of the basic goal here. So for proteins in particular, I was talking about sort of SE3N. So this means basically a space of rotations and translations. And so here you represent sort of protein as a long string of beads where each one has a rotation. And what's nice is this is sort of very good representation here because you're sort of able to represent very rigid lengths, but also sort of very independently. So there's many different representations here. This one's nice because it's sort of independent between residues, but also you can sort of build into the known rigidity between atoms within a residue. And so we're sort of able to leverage this nice representation for these manifold spaces. Of course, this presents difficulties for sort of diffusions and flows. So for sort of flow models, this is very nice because you're able to do things on manifold very easily. And what I mean by this is sort of flow models, you only need to know sort of where you're going and where you're starting, and then be able to sort of direct a line between them. And so this sort of, right, this math basically says that, well, your sort of conditional velocity, so conditioned on some z, which is in this case 2 noise points, is just sort of a logarithmic transform, so sort of the direction that you need to go. And so the sort of the intuition here is basically diffusion. You would need to figure out sort of how heat diffuses over a manifold, which is very hard for sort of arbitrary manifolds. But for flows, you only need to know sort of the direction that you're going. And so to train these sort of models, basically, you need to train your velocity field to match this direction. And the way that I think about it is sort of, you have this like Earth or something, right? The sphere, of course, SO2 instead of SO3, but same idea is basically you're trying to build a model that says which direction the plane is going on the sphere. And so this is sort of the basic idea here. And that's why, sort of flows are useful. And one thing, we found here is that you sort of, if you train these models, they underfit in very specific ways. So in particular for this type of model, when you do sort of both rotations and translations, it turns out when you sort of, when you just train it, you should be able to sort of just do standard inference, everything at the same time. But if you do that, it doesn't work very well. And so, or you need to train much longer. And so if you train a long time, it works. But if you train not long enough, basically, if you multiply the rotation speed during inference, you get much better results. And so what does this mean exactly? So even like these sort of models that I was talking about, also the diffusion models, it turns out this is the case too. It's sort of something subtle about our architectures of our models that if you're able to speed up the sort of rotations first, then it's much easier to do these unconditional design problems. And so, why is this the case? It's basically what we think now is that it's sort of determining the secondary structure before the actual positions. And so, it's sort of like there's quirks about your specific data types that make it easier to learn one thing versus the other. And so, I guess the sort of space of generative models, it matters sort of how you parameterize things and how you control, right? So here we're sort of saying, maybe you do rotations first and then translations. but in other spaces it might be different. And so, all these protein design things, well, there's some problem here. I was talking about sort of flow model where you go from sort of a noise to a something that looks like a protein. You go, but that's not the sort of whole design process. Most of it is sort of this filtering step afterwards. And so, what generally people use is once you create something that you think looks good, you create a bunch of sequences for it, and then you refold those sequences to get another structure, and you compare the two and say, ah, yes, if these look the same, then it might be good, right? It's because sort of you have three models agreeing that this is the right thing, and that's better than some random model, I would say. Although this has sort of many problems, right? So in particular, right, three different models. This is not sort of end to end like we would like, but it's sort of a good filtering step. And so I guess some problems with this, right? So this is sort of where I'm coming from. Is like, well, we want to do protein design, but we're just making the things that we already know about. We have to filter them down a lot. This is sort of a big problem. And so sort of issue one is sort of diversity. So this sort of procedure where you filter a lot, you basically, only get things that agree with all models. And so you both sort of limit the diversity of your model, but also you have to filter through many things, and so you get a sort of very small subset of the possible space. And so there's sort of one thing that's a big problem here. The second issue is sort of dynamics, right? So proteins move a lot. I didn't talk about this much, but basically all the data that we have is very static. And so you're trying to, sort of figure out, right, proteins are actually moving. And so you want to understand this sort of dynamics, but that's also not in our data. And finally, the third thing is sort of cost, right? So there's many different models to do this, but most of them basically you design many, many more things than you actually are able to test. And that's of course sort of okay, of course, because compute time is much cheaper than experimental time, but it's really way more expensive than you would really need, right? So, in particular, right, so BoltzGen, right, is a recent model. Of course, there's sort of many problems with it and many nice things about it, but here's sort of the numbers, right? So you design, basically 10,000 or more different designs, and then you test only, 10 to 100 basically designs, and some of them work, right? And that's great, but of course, anybody that sees, well, really only, what, 0.1% work, right? We can do much better. And so I guess the question is sort of, how do you improve this pipeline? I think there's many different ways, but sort of from a modeling perspective, right, where can you do the best by improving the modeling part? And so I'm going to sort of claim there's two different pieces you need. So first is sort of more controllable generation. This sort of helps because, right, the current way of doing it is generating a lot of designs and then filtering it down. If you sort of are able to steer your generations to be better, then maybe you don't need to generate so many. And then the final thing is sort of, only some of our things works, right? And so how do we build in sort of better information to make it more likely for sort of something we think of the computer to work also in the real world? So yeah, that's sort of the framework I'm going to talk about. Is like, we have these generative models; they're very powerful, but if we would like to steer them, if we would like them to be sort of more successful, how do we do that? So, sort of stepping back, sort of in generative modeling in general, right? In general, we just have this big data set as big as we can get, as diverse as we can get, so that everything is in distribution. We build this model, right? A generative model that says, let's just learn from all this data that we can, mostly by just adding noise and sort of trying to recover the noise. And then doing inference to sort of get the bias back. And so basically here, we're trying to, it's sort of uncorrectable in some sense, right? We're not able to, once we have the data, once we have the model, you can't fix it. And so sort of what I'm thinking about is ways to fix it. And one of those is sort of have some other function that says what is good and what is bad. Here I'm calling it energy, but there's sort of many names for this, right? You could call it a reward function related, right? So, posterior, right? There's many ideas about what is a good way to say. And really, of course, this is different than data because it says, here's something that says what is a good data point and what is a bad data point. It's like a function that says, here's what I want, here's what I don't want. And so oftentimes it's much easier to obtain than getting, the data that you would like, basically. And so the idea here is how do we sort of combine these things, right? So, most of these times, right, this you can define this function, but it's very hard to sample from. And it has this sort of intractable normalization. So if you're able to sample from it, that would be great. But we can't really get this, samples from it. We can only get sort of evaluated on things. And so, the classic way to do this in proteins or molecules, right, is to do molecular dynamics, or in other cases, more general MCMC methods. And so you can take an energy and get data, but there's sort of many problems with this. And in particular, it's very slow, right? So depending on your space, it can take a very long time to converge, but also if you have a different system, doing MD in one system doesn't necessarily help you do MD in another system, something like this. And so, one way to think about this is to sort of train models directly from your energy, build a model, and then do inference. That would be great. And in fact, that works sometimes, but it's quite hard to scale. And so if you have some data, I would say that's the sort of best case where you were able to sort of train a model from data, but also have energy and then control it to what you want. So really, I'm trying to consider sort of models where you're able to sample from sort of some distribution that you can define, right? That you can actually, specify what I would like to sample from, and then build a, better data set, right? Sort of something that you would like to be sampling from, and sort of build quick samplers that are able to, given a new setting, here's what I would like. And so that's sort of what I'm going to talk about, is like this paradigm where, we sort of first talk about some different types of flow models, then talk about sort of diffusion samplers and how you go from energies to models, and then how you sort of do guidance with you have energy and you want to sample from something, and then talk about sort of Boltzmann generators, which is one sort of specific application in molecules. So great. So yeah, so normalizing flows, right? These have been around for a very long time in the generative modeling paradigm. Basically, you take a very simple distribution that you can sample from, you apply some invertible transformation, and then you can get likelihoods, right? So this is quite nice, and you can train with maximum likelihood. We sort of abandoned these a long time ago because they didn't scale as well, but it turns out sort of more recently they're able to work relatively well. The big problem here, right, is that you need sort of invertible architectures and a very structured Jacobian, so that's very quick to evaluate. And so this is always the problem with them. It turns out with more powerful networks, it works okay. And of course, right, you can make these deep, right? So you can just stack a bunch of invertible layers. It's still invertible. You can sum all the log determinants, and you can still get the log likelihood, and that's great. But one thing that's very interesting that happens with sort of infinite number of steps, right? You get this continuous domain where you're able to transform. Sort of, it's now an ODE instead of a series of invertible transformations, and that allows you to sort of do this nicer formulation. And so what's very nice is that the inverse is sort of for free, so you can use any architecture you would like, but also get the inverse of it by just taking the negative sign. And so this is sort of a nice way to go if you need likelihoods and are able to steer things. So what does this look like, right? If you're trying to transform distributions, you sort of get this very smooth velocity field, and you're trying to understand, how do you change things over time. But sort of still the problem here is this likelihood calculation. So if there's sort of so this is sort of the trace of the Jacobian. It turns out basically to evaluate this, you need sort of to evaluate you need sort of the whole Jacobian. That means you need to evaluate your function d times, so dimension number of times, and so that's quite inefficient relative sort of generation. And so yeah, diffusion models, right, are very similar to sort of continuous normalizing flows in the flow matching paradigm now. The only difference is sort of the stochasticity. If you do sort of the noising process, you add noise with possibly some drift, although it could be just zero, and then you try and reverse it and learn the score, right? But the interesting thing here is, I'll talk about a little bit, you can estimate the density change quite efficiently, although non-precisely. And so it's a sort of a trade-off. So you get some things about with noise that you can't do with just the deterministic ODE. Yeah, there's sort of different types of flow models, right? There's many ways to go from sort of map a noise point to a data point. And what's interesting is there's sort of different ways to get likelihoods, and I'll talk about sort of why that's interesting for control. Yeah. And so yeah, okay, so flow models. Now talking about sort of diffusion samplers. So right, molecular dynamics is nice, but it takes a really long time. And so you need these very small time steps on these sort of very bumpy energy functions. You get these large simulations, and then you need to start from scratch every time. And so one thing that's very nice about sort of our diffusion and flow models now is essentially you can think about it as you take your data, you add noise to it, and you sort of smooth out the energy landscape, right? So this is one very nice way to think about smoothing the energy landscape, and that would make it much faster to do sampling. And so yeah, this is exactly sort of what the diffusion models do. If you just sort of take your energy function, whatever it is, if you add enough Gaussian noise, it becomes Gaussian and very easy to sample from. And so that's the sort of basic idea here. So I'm going to ask sort of 3 questions here about how you build sort of samplers in general, right? So can we sort of train a diffusion model if we only have access to energy and not data? So diffusion models are generally trained sort of on these conditional scores, but we would like to train it from energy, not data. And then can we sort of leverage temperature, right? Temperature is an interesting way to sort of smooth things out and makes it easier to learn. And then finally, can we sort of leverage these sort of temperature and diffusion together to get these sort of very smooth things to train in sort of larger scales? And so, we talked about this sort of diffusion models, the stochastic flows, right? But this sort of way to train it is based on the score function, and this requires data points, right? So you need to sort of sample data points, calculate the conditional score, and then do a regression problem. But if you don't have the score, then you have a problem. And so how do you estimate the score, right? It's via convolution. That's you take the data, you convolve with a Gaussian, and sort of smooth it over time. And so learning this, right, we're able to sort of, you can't directly evaluate the marginal score. Instead, you only have the conditional score, right? As I was saying, and you sort of need to just solve this regression problem, right? But of course, the first step here is to sort of first sample from a data point, then add noise to it, and then do this regression problem. And so this first step is the problem, of course, if you're only trying to do an energy. And so, yeah, how do we train it with just an energy and not data? And so, yeah, this first part is the problem. And so what do we do about it, basically? So the sort of way to think about it is that, yeah, currently you sample sort of a noisy point from your data. And you can think about this, right, as a convolution between your data distribution P0 and a normal distribution with variance sort of sigma t squared. And if you're looking at the score, which is what we want to learn, this is the formula for it. But then what you do generally is distribute the gradient inside of the normal distribution, and that gives you the standard diffusion formula. So you sort of are able to sample from P0 and then take the gradients of the normal distribution. But you can do it the other way too. So if you distribute the gradient to the data distribution instead, you get a different formula that can also get you at the score. And so this is sort of one way to think about it is if you distribute it the other way, then you get a different formula that is a different estimator of the score, right? So what does this estimator really say? Right. So here you sort of take any xt as input. You sample sort of noise, sample normal distribution around that towards x0. You evaluate the energy on it, and then you take the gradient, the log sum x of it, and that gives you a different estimate of the score. And so this we call sort of denoising energy matching, because you're sort of matching these denoising based on the energy function, and this allows you to give an estimator that is based on just the energy and not the score, right? So what's interesting about this is it's able to train based on an energy, but the problem, of course, is like this estimator may be unstable, right? So the sort of standard diffusion model sampler is perfect, but this is an approximate. So the problem here is basically if you take a log sum x, this is biased unless you have infinite number of samples, and so we're constantly sort of fighting this bias if you're learning this way. Yes, please.
Just to give you some time to breathe.
Yeah.
I guess your data distribution is not something real, right? It's something that you made up.
Yes.
Putting some Gaussian noise into it.
Mm-hmm.
And I guess, and I'm just thinking about molecules because I don't know how to think about anything else. When you noise molecules in the Gaussian way, that doesn't necessarily mean that they're real molecules or that
they're definitely not, right?
So I guess, I just have this philosophical problem. We have the same question in our lab where, people sort of talk about these scores. And they're like, oh yeah, and I took this score from this trajectory, but then I don't really know what it means. I don't know if it tells you more about the optimization process of your sampling, which basically tells, well, I can get to this solution kind of smoothly and kind of easily, whatever that means.
Mm-hmm.
But does the steps that you take in between from A to B really tell you something about B itself?
Hmm. Yeah, I mean, generally not so much, right? I think because so most people try and take xt and then estimate where you're going to go, so get an expectation of your predicted point. But that's that's sort of doesn't look like a molecule, basically, because it's sort of like an expectation of all possible, sort of your distribution of points where you're going, basically. And so I think I guess sort of a very recent advancement is sort of these few step models. We're able to jump in not many small steps, but sort of 1 or 2 steps. And that's very interesting because it allows you to consider sort of better estimates of where you're going. And so if you're doing that, then you could sort of map back to your noisy space and figure out where you're going in sort of a better way. And so then you're sort of looking at, where is your molecule going in sort of molecule space rather than sort of this like noisy, weird space, I would say. But yeah.
Then if you actually compare estimations of those energies from your model to, let's say, controlling something control or solving like some other energies, either being like statistical or more physics based.
Yeah.
They tell you similar things or different things or
so I think this so most of the time, right, the smooth energy functions you would do like based on temperature or something, right? And I think you can compare it to these kind of things, but I think it's quite different. And it's much easier to learn these kind of things, right? And so it's sort of the trade-off I feel is like Gaussian noise is the best thing to learn at dimensions, but we have no idea how to interpret it. So yeah, this is the trade-off, right? Yeah. Okay. Yes. So basically how this works is we sort of build the data set over time, sort of this replay buffer, and then use that to figure out where to train, and then use this as an estimator and do it over and over again. And so still, basically the idea is to sort of build a good data set using the sampler trained on the energy. And so yeah, this was the sort of first idea about how to build these like simulation-free diffusion samplers, but you're only able to do it on sort of small things, right? So this is very easy energy, in particular in sort of molecular cases, but it still is very sharp, right? It still like has terms to the 12th, distance to the 12th. But that's a very difficult energy to sort of sample from if you're just doing MCMC. But if you're smoothing it out, it becomes very easy. And so this is the question is really, can we leverage these like diffusion type things to smoothen the space and be able to control it to where we want to go? Yeah, so I guess the takeaways from this part, right, is like, can we train these diffusion models? Yes, of course, but the problem is it's quite difficult in terms of the number of energy evaluations, and it's very hard in sort of higher dimensions, right? So we tried bigger things and through molecules and sort of got stuck there. And so, yeah, I guess the question is, right, like, can we leverage temperature? So this is another sort of direction is like, right, we were just talking about sort of diffusion sampling, which is smooth as one way, but this is sort of a different way to smooth based on, so this is beta, which is inverse temperature, and we want to smooth it out this way. So, just sort of intuition about why this is a good thing, of course. If you smooth it out, how does it help? Well, if you think about molecule space, you sort of get much more coverage, right? Much more smooth energy landscapes. This is sort of a Raman plot for just like this one angle. But really, of course, it's in general much easier to sample from sort of these things with smaller gradients. Yeah, please.
Understand. So here you still do score matching in the sense that you use the divergence as the loss, like as you showed.
Yes.
But you parameterize the energy and not the score.
So we haven't done that yet, but we will. And so there's sort of you can do that, but
then you get a second order derivative.
Yes, exactly. You get very ugly stuff. And so, yeah, I think this is one of the hard parts. I'll talk about this in a second, is like, how do you train sort of energy functions instead of score functions? And currently our methods are very poor for doing this.
So then when you say that you have denoising energy,
yes,
denoising score matching.
Yeah, we call it energy because it's from an energy function instead of from data. But yeah, it's not exactly, you don't learn an energy function. You still learn a score, but it's from.
You can use the score to generate new something.
Exactly. Yeah. Yeah. Okay. So if we want to sample from a temperature nil distribution, so actually people in sort of many domains try and do this, right? And the classic way is just to sort of scale the score, right? So if you scale the score by beta, what do you get, right? So this first thing is true, but if you just do this sort of, if you learn a diffusion model, you scale your score by beta. It turns out you don't get the right marginals. And so this is sort of a nice trick. It sort of works. You get like something that looks sort of temperature annealed, but it is not exactly the correct distribution. And I'll sort of tell you the difference in a second. That's all right. If you have these diffusion SEs, you're just moving particles. Diffusion framework in general, the density changes like this. So it's just the Fokker-Planck PDE. You sort of get the score goes to a diffusion components, and the same thing goes down. But if you put everything to a beta, these are no longer equal, and basically you get one extra term. Like, so this is sort of a reweighting term. Basically, you need to take your PT and then you also need to track weights. And so this is the additional part, is essentially when you want to simulate sort of the correct thing for a anneal distribution, you also need to add these weight terms. And so you need to simulate a weighted diffusion SDE. So if you add the beta here, the correction term basically that you get is you learn weights, which HT exactly, I'll talk about the form in a second, but it's sort of some other function of X. And that allows you to do if you simulate this equation instead, then you get the correct anneal distribution. And so what's interesting here, right, is that you sort of need to do one extra step to get these correct distributions. This reweighting in particular is very useful. Let's see, how does this look like, right? So normally we sort of take our initial particles, but here, so the first thing we do is we compute these weights. We resample the particles, and then we move them around, and then we do it again, right? So what does this mean? It's sort of like diffusion, but you're adding one more resampling step, and this allows you to sort of do a correction to the correct term. And so yeah, for this annealing in particular, this is what it looks like, right? So if you just do sort of scaling by the inverse temperature, then you get, like, if you're just doing from a Gaussian to 4 Gaussians, you get something that looks pretty correct, but is not quite there. And so you get these, right? If you do this, you sort of move particles, you scale the scores, and then you compute this resampling. And so this HT, in this case, is sort of this is sort of the magnitude of the score, and that's sort of the right reweighting. So basically, you upweight things that have high scores, and you downweight things relatively if they have smaller scores, and that allows you to get sort of the right distribution PT to the beta here. So it's quite interesting for sort of different ways to do this, and this is all connected to likelihoods, and I'll show you in a second like why that's the case. And so I guess the interesting part here is, like, once you have likelihoods or some estimate of likelihoods, and you're able to control things better. And so what we showed with these is basically if you're trying to train these sort of sampling methods, it's easier to first sort of train at a higher temperature and then anneal it down, rather than directly trying to train at a lower temperature. And so you can sort of see if we start train this model at temperature 2 and then try and anneal it down to different temperatures, if we use this sort of FKC resampling stuff, we're able to get less error than if you don't. And so, like, looking at temperature 0.8, these are sort of the MCMC data set, which is the true one, the green one. And if you do FKC, it's closer than if you just try and directly train at temperature 0.8. And so this is quite interesting. It's a different way to smooth, and enables you to train at a higher temperature and then anneal down. But yeah, so I told you this sort of requires density, which is quite interesting. So the real question is, like, where do these densities come from? And so, in the sort of flow match sort of ODE case, right, this continuous normalized flows, you start, right, you sort of try and figure out how the density changes over time, right? And so how does this change? It's basically, there's two components: is how it changes in time and how it changes in position. So this is the general, like, hard to evaluate term, which is the trace of the Jacobian. But this is sort of annoying and only works in the ODE case. If you have the SDE case instead, then you still need to write this term, which is the sort of change in density, but you have this extra noise term. And so in particular, you sort of have the sort of diffusion term, or the sort of diffusion, and so that's sort of due to the noise term. And so, but one very interesting thing happens is sort of if you apply this Ito's lemma, you can sort of I guess the right way to think about it is, like, there's going to be a cancellation here that tells you you could sort of cancel out this very ugly second order term, and it'll get you an interesting estimate of the score. And so basically what you get, I'll skip some. So yeah, the problem is sort of this Laplacian term, right? Normally you can evaluate an ODE, but if you use an SDE, you can cancel it out and get a very fast estimator of the density. And so that's the whole idea with this Ito density estimator. If you collect everything together, you get a different estimate of how you change likelihood. And so here we have no complicated terms other than a score. So F here is like is either 0 or something like -x, so something very simple. And you can sort of, for anything, you're able to evaluate the density if you have the exact score. And so yeah, you can use both sort of the vector field that you're transporting with, but also any vector field that you would like. And so in sort of guidance task, this is sort of much faster than the density estimate that you get. And this is actually sort of the math that we're using to calculate the correct weights for the FKC stuff. And so this is very interesting because I didn't know you could do this. Basically, there's sort of a new type of density estimation for SDEs, and that allows you to sort of do better control, even if you just have this estimator. And so it's sort of with very little extra cost, you're able to use sort of likelihood-based control for scores. And just some notes, right? So this is sort of a very nice principled approach mathematically. It works with any architecture, sort of any number of models. But the problem here is that for the exact density computation, you need to the exact score. And so, of course, we're using learned scores instead. And so then we sort of accumulate these biases. And so what we found is sort of very close together. It's a great comparison. But if you're looking at very different things, the sort of you sort of unsurprisingly, right? Like it gets uncalibrated. You get very far away from each other. So if you're looking at image space and you have two sort of close together images, it works very well. But if sort of relative densities work, but if you're looking at very different things, it doesn't work very well. And so yeah, this also applies to not just temperature, right? So if you're looking at like to control, for instance, for different properties, right? So if you wanted to say, okay, proteins, for instance, if you have binders to protein A and protein B, for instance, and you want to do sample something with both, then you could do sort of, right, if you sort of want to do this inference where you get both properties at once. And so the standard way to do this, right, is sort of averaging scores again. So that just, you take both models, you add them together, works, fairly well, gives you something in the middle. But there's sort of many other ways you could think about is mixing these densities. And so, a couple that we consider, right, is like mixtures of densities. You just want either to sample equally from either density, or one that's interesting is if you want them to be equally likely. So you sort of look for points that are both A and equally likely to be from A or B. So that gives you sort of these lines in the middle. And what does this look like on sort of images, right? If you take a stable diffusion model and ask it for these prompts where you're prompting for two different things, you get these ideas where, you sort of like, you can't tell, should it be a bird or a tree or, something like both, right? So it's sort of one way to think about equal. Of course, there's sort of a different way of mixing, right? So annealing, I talked about a little bit. But also thinking about products, right? Products are very attractive because it's sort of the natural way to think about having property A and property B. And so we'll explore a little bit about how you do that. And so, so I guess so this is work by Marta, who's presenting at AMLD right now. And so she's interested in sort of structure-based drug design, so small molecules against protein pockets. And so, what if you wanted to design towards sort of two proteins at once? And so, you can do it's basically exactly the same math. All you get is a different estimator. Sorry. You get a different estimator of the reweighting, and you sort of average the scores. So, we take these sort of standard formulas and how we mix things together, but then we add this resampling step to really correct it to the correct thing. So, if you just do averages, it works. It gives you something that's in the middle, but it's not quite correct. And so, if you do the resampling, then you get the sort of correct product distributions. And so, we can show that it works slightly better if you do these correct things. So, we're looking at docking scores here, and so we have two different targets, and if we do sort of compare average of scores versus FKC type methods, so basically just adding resampling, then you can get slightly better on sort of all the quality metrics, but you sort of get slightly less diversity. And this is because of the resampling. If you sort of take more samples to get sort of the same thing. So it's sort of a trade-off here, but I think in general, we're interested in sort of higher quality things. So could be quite useful for that. And so, this is an interesting idea about how to control distributions. Basically, you're able to, sort of take these likelihoods, these or very approximate likelihoods at least, and sort of correct the distributions even in sort of more practical settings where, it's sort of where likelihoods we think were very useful in small dimensions, they also can be used in sort of these medium or large dimension settings. So for this, right, we're able to leverage sort of smoothing things or, other distributions to sort of make things easier to learn. But right, I talked about the likelihood estimator is only biased if it's perfect. And so this is a problem if you want to do big jumps to temperature, right? So we jumped it from 2 to 0.8, but if you jump really far, sort of accumulate this bias and you're not able to go further. And so, the question is, so the final question is sort of how do we scale systems by leveraging both the temperature and diffusion. Yeah, sort of, Binner was talking about sort of how you talk about different ways of like smoothing energy functions, right? So diffusion is like this, right? Like it's moving particles. I think about it as horizontally, right? Like sort of smoothing out that way, where temperature is sort of different and sort of reweighting between modes to make it smoother overall. And so sort of like two different ways of smoothing out the space. And so there's sort of methods for sampling for both types of things, right? You take diffusion type samplers, many different models, also many different models if you're trying to do annealing based processes. But really, of course, there's, how can we combine these, right? There's sort of different ways of thinking about smoothing and with different advantages in particular. So there's sort of two different things about them. So one question is, one thing's nice about temperature annealing is that you can sort of evaluate any of the densities along temperature, whereas diffusion, we have no idea really how to, given an X at some specific time, evaluate the density there, other than, well, you need infinite convolution basically in space, whereas temperature you can. But the interesting part about diffusion is you're able to learn these sort of smooth vector fields, which is not true about temperature annealing. And I'll show in a second why this is the case. But this is sort of really one of the nice things about diffusion is it's much easier to learn than sort of different types of annealing. So thinking about temperature annealing, let's take the same example, sort of two Gaussians, and thinking about sort of velocity to evolve over time. And so this is sort of what the vector field looks like as you evolve this, as you sort of sharpen this density into the two modes. And so what it looks like is you get this sort of sharper and sharper, right? Sharper and sharper distributions, but also really spiked in the velocity field, right? And if you think about why this is the case, it's really because there's sort of more mass in this mode and less mass in this mode relative. And so what's happening is you're trying to shove mass from one mode to the other. But this is a very thin tube, right? So when you're trying to shove a lot of mass to a very thin tube, the velocity needs to be very high. And so you get these very spiky velocity fields, which is something that neural networks really do not like to learn. And so this is the problem as we try and like learn these models that are trying to deal with temperature. You get these very spiky sort of fields, and then you're not able to learn them very well. And so, yeah, sort of this relative weighting between modes is not preserved in temperature. And so you sort of need to, think about how you mix these two things, right? Temperature is very nice because we can evaluate the density, but it sort of results in these very exploding transport vectors. Cool. And so, okay, so this is the idea about how you might want to get around this. So if you had samples from very high temperatures and you would like to sample from lower temperatures in an amortized way, one way is to sort of train these models iteratively. So the first thing we do is collect MCMC samples at a sort of high temperature and then try and anneal it down to lower and lower temperatures. And so you train these score models and an energy model at the temperature, and then try to anneal it down. And so you sort of take a Gaussian, learn this sort of standard, sort of take the data, right? Now we have data from MCMC, because we assume it's high temperature and much easier to sample from, and then try and anneal it down. And so using this sort of inference time annealing of temperature, we're able to take samples from the Gaussian, use our complicated derived weight equations to get the correction. And so now we can correct down to a lower temperature, which then you can train a model at the lower temperature and do it again. And so this is quite interesting is because you're able to sort of like debias, sort of do this like annealing over time and temperature, so diffusion and temperature, and then get these sort of increasingly nice samples from higher temperature. And so this is sort of an interesting way to think about how you do this annealing, and we call this sort of progressive inference time annealing, because you're able to progressively anneal from high temperatures to lower temperatures. And so one thing that we did here was train a score model and an energy model. So you were talking about sort of like, do you train an energy model or score? Here we train both. And the nice thing about training an energy model is you could actually get this reweighting sort of with no bias. And so it sort of gives you relative likelihoods, and then you're able to do resampling. And so if we were able to train these very well, I would use them in sort of everything. But the current problem is training energy models is very hard. So when we train these, basically, we spend all our time trying to train the energy model well. The score model sort of directly trains in like, maybe 2 hours, and then we train like another 48 hours to try and make the energy model work. And so we basically, so this is the best way we got energy models to train in sort of this type of setting. And you can see, I guess, some interesting things here. So, okay, so how does this work in general? So basically, first we train a score model the standard way. That works very well. The second thing we do is sort of do this this matching. So for this from sort of the score to like the targets. Like so here we pretend we also have energy basically, and so do this target score matching, which allows you to train from the energy also. And so that sort of smooths out if you have the energy. And then we have this energy distillation, which says take your energy model, take its gradient, and match it to our score model. So if you try and sort of train the energy model directly, which you can do, it doesn't. It's very unstable. And so this sort of helps it give you a consistent target. So this, right, it sort of removes the variance of this denoising score matching objective. And then the final thing to do is like pin it to time zero. So pin it to match our energy function. And so this is sort of the way that we figured out sort of how to train it. But of course, it's very complicated and very annoying to train. But yeah, we sort of added many special tricks to make this work. So as many possible tricks to work. So exponential moving average, all the special parameterizations that we could find. So this is parameterizations from EDM. Clipped everything you could possibly clip to make it more stable. We make the sort of sigma min large. So this is sort of the lowest amount of noise. So it turns out that these things become very stable at sort of low noise regime. We add lots of compute, make it 10 times bigger, and also like do a lot of tricks to make it 10 times faster, and then use all the preconditioning stuff from other models. And so this is sort of like the most stable we could get to train, I would say, is sort of small molecule settings. And so we showed this on alanine tripeptide. So this is the biggest you could get essentially with no data, as far as I know, on molecule settings. And you're able to sort of do at least approximately correct modes, sort of given the same number of energy valuations, you're able to get a similar, sort of better than MD. So this is sort of MD and then diffusion. So you should compare these two. Basically, that's sort of the simple baseline where if you just ran the same amount of MD and then train a diffusion model, you would get similar results. And so yeah, that's the sort of, I guess, one way to think about, right? If you really want this sort of consistency, you pay a lot in training these energy models, but if you do, you can get slightly better results than if you sort of didn't use these. But so yeah, I think this is one of the big open questions is, we can train all these flows very easily, but if you really try and train these sort of energy models where the gradient is equal to flow, you have massive problems. And so, I think this is really still a problem in the modeling side, right? It should be possible. It's sort of a gradient problem and a learning problem. Although, yeah, that's sort of the sort of state of things today is like, I really would like to train these energy functions because it gives you sort of likelihoods and control, but we're not able to do it at high scales to really be able to do what we want. So that's one of the big things to work on. Okay, so one last thing is sort of, what can you do if you also assume lots of data? So all those models are assuming really, no access data or data from only high temperature. What happens if you also have a reasonably good data set? And so this is sort of the realm of Boltzmann generators, where you train a sort of flow model on your data, and then you reweight it to your target distribution. And you sort of need some things for this, right? So you need some way to sample training sets, and you also need to be able to correct your model. And so this is the sort of correct the error in whatever model you get and resample it. And so this also requires basically likelihood to remodel, and you're able to resample it. So yeah, sort of you need a likelihood-based generative model, and you're able to do self-normalized importance sampling to correct it to your target distribution. And so you just do this reweighting where you take the target density over your model likelihood, and you're able to get consistent samples. And that's your importance weights. And then the big problem, of course, with all these reweighting schemes is if your variance of your weights gets too large, then you get these collapses, right? So you may end up with many fewer samples or something not very useful. And so yeah, the big question for these is sort of, you need generative models of fast inference and fast likelihood computation. And these sort of are much, there's much less work on this than I would like. So, for flow matching models, they're very nice for sort of training, but the fast inference is a maybe. The fast likelihood is a definitely no. Diffusion models, you can get sort of nice sort of training also, but again, you get fast inference, but really no commutations that are exact enough for sort of normalizing flows. This is sort of a reasonable train. They're really hard to train, but once you have them, you get fast like inference and fast likelihoods. And so I guess the question for me is like, how do you sort of merge these two things? And so for this, we generated a lot of MD data to test like transferability between peptide systems. So this is 22,000 just randomly sampled peptide sequences from like 2 to 8, and simulated with a very simple force field. And we selected these because we wanted to figure out sort of transfer between systems, how well it worked. And then this is all publicly available, of course. And so the model we trained is called basically based on the tar flow. So this is a normalizing flow-based parameterization. So this is very nice because it's fast for inference and fast for likelihood computation. And sort of recently, basically, these were shown to scale to ImageNet type things. And the only big difference between this and what people were doing in 2018 is just adding this, block in the middle that's called a causal transformer, and as we all know, works quite well. And so I guess for this, right, you can calculate the log likelihood quite easily, and if you need these sort of permutations. And so what does this give you, right? So if we take this reference MD set, this could take a lot of energy valuations, but then we compare it to what we do in sort of much fewer energy valuations and sort of a shorter MD chain. So here, this MD doesn't mix; it's stuck in one mode. But what's interesting is the generation sort of finds this mode that our long MD hasn't captured, which is quite interesting. We're like, oh, what's happening here, right? So and what we find is basically if you run it for 50 times longer, then this mode also appears. And so it's quite interesting. Basically, you can given this sort of very transferable model, you could sort of find modes that you weren't able to before. Sort of using medium scale MD. So you need sort of very long scale MD to find these types of modes. And so what I find very interesting is it means that we're training on sort of very short MD chains, but many of them you can transfer between sequences, right? And so what's probably happening is this mode exists in some other sequence. We don't know which one, right? There's some sequence with that mode with much higher probability, and then when we transfer between sequences, we're able to find sort of for a new sequence. Maybe it's much less likely and hard to find with MD, but since we've seen it in other sequences, we're able to find it very quickly with these sort of transferable models. And so I think this is very interesting, sort of way to think about it is like there's sort of two different ways of thinking about, finding different modes. There's sort of things that are more and less probable in different sequences, but also, sort of faster ways to explore a single model. So, and so, the thing we're interested here again, right, is like given the same number of range evaluations, how do you perform? And so we leave out, a bunch of test sequences, and we see, well, given the same number of energy evaluations and a fixed compute budget, you're able to beat sort of the MD. And in particular, right, especially on these metrics that measure sort of how many modes you've covered, and so you're able to sort of cover the modes much better than sort of equivalently length MD chains. And so sort of the takeaways here is like Boltzmann generators, they scale to much larger than we were used to. We were able to scale before. Sort of 200 atoms is sort of how big we've done so far. They transfer surprisingly well to unsieved systems. The importance sampling works much better than I would have thought in these high dimensions, right? Classically, it was that sort of importance sampling fails like exponentially fast, and then these normalizing flows work quite well. And finally, I think we can retrain sort of even larger models. So these are still very small peptides. To make them useful in proteins and dynamics, you really need to make it work more. So we've been trying to train sort of, this is a model that we just submitted to ICML, sort of models based on just language models instead of normalizing flows. Since there's no normalizing flows at all, we're trying to sort of do the same idea. And this, you could get sort of these nice scaling curves if you use just causal transformers, basically. And so this sort of generates fully autoregressively and could do even better, significantly better than the sort of our PERS model on big things. And so this is, sort of what I'm very curious about is like, are there different types of, models with likelihoods that we're able to do this resampling quickly and get sort of efficient sampling, but also, transferable across different systems. So, so just summarizing, right? Like, I think, if you want to draw these sort of consistent samples, you need these if you want to keep getting better and better data, you need to be able to correctly sample from whatever sort of proposal distribution you like. And so this requires like likelihoods or resampling or something that gives you a way to sort of steer where you would like to go. And that really requires sort of efficient access to these likelihoods. And so really trying to build models with better likelihoods that are able to sort of improve our data set over time. And then, yeah, these sort of PROS and PDIP methods sort of, how to sort of build these controllable generative models that are able to improve, and given an energy, right, given something that we know we would like, please give us something better. So yeah, that's sort of the end of my talk. Thanks very much, and happy to take any questions.